All right. We're going to talk about blood typing. This is to prepare you for the lab that you will be doing this week, whether it's in the cafeteria. If you're in periods 2 and 3, you'll be in the cafeteria without me, so please pay close attention. And you guys can work together, and you should be able to figure out the lab pretty well just with each other and the packet. If you are in period 10, you'll have me, so it's not too big of a deal. If you're taking the Keystones, you will come to the makeup lab on Thursday afternoon for the PM activity period, and we can go do the lab then, but I want you to go through this so you understand what's happening. So with blood types, you've probably heard of blood types before. There's type A, B, type AB, and type O. Oh, hold on one second here. All right, sorry about that. I wanted to get my pens up here. So you probably have heard of these, A, B, AB, and O. These are your phenotypes, which means a physical characteristic. You have these different types of blood, meaning your red blood cells have markers marking them by these different, they're actually called antigens. We'll get to that. Your genotype then shows your alleles that make up your, the alleles that you have. So you notice for type A, you can be capital I with a superscript of capital A. You can have that twice. That would make you homozygous dominant. The thing about blood types is that it's a co-dominant trait, which means the capital I can share its dominance with another capital letter. You'll notice there's also a lowercase I right here. The lowercase I is recessive. So type A blood, if you have the big I superscript A with a lowercase I, you're also type A blood. Type B can be I big B, or I big B lowercase I, because again, that's recessive. They would only have type B blood. Type A would only have type A blood. Then there's type AB, because it's co-dominant, you can have both letters, I superscript A, I superscript B. If you do, then you have both A and B markers on your blood. Lastly, there's type O. This is the recessive trait, homozygous recessive, 2 lowercase i's. They actually have no markers on the blood. Something we've talked about before, why are there 2 letters for each genotype? You should know this. We get our genotypes from our parents. So each letter, each allele comes from a parent. One from mom, one from dad. So if you notice the first example for the type A, the first genotype, that means both parents passed on capital I superscript A to make that genotype. One thing to note, a person has blood cells that correlate with both alleles. So if you are type A, you will have, if you're this first example, the homozygous dominant, you will have all the same blood cells. If you're the second one, the heterozygote, you will have blood cells corresponding to the capital I superscript A. You will also have a few blood cells corresponding to your lowercase I. Same with the type B. If you're heterozygous, you will have blood cells representing both alleles. Type AB, you will again have both represented, and type O, since it's homozygous, doesn't really matter so much. All right, the physiology, we've already mentioned antigens and antibodies. Antigen is a foreign molecule, as long as your body knows it's not marked as self, but be considered a foreign macromolecule that doesn't belong to the host organism and elicits an immune response. The antibodies are antigen-binding immunoglobulins. They're a protein that are going to bind to the antigen. It's produced by the B cells, and it functions as the effector in the immune response. What does that mean? It means that when there's an antigen in the body, as long as it's a foreign antigen, meaning your body doesn't recognize it as self, that's bad. And an antibody will come along and tag it as bad. Remember, the antibody doesn't actually do much itself, but it'll tag it as bad and cause the immune response to occur. So what does this mean for blood types? A person with type A blood is going to have markers on their blood cells. Remember that all cells have antigenic markers, meaning all cells are technically antigen, It just depends if your body recognizes it as a self-antigen or a foreign antigen. So notice this person here, the homozygous dominant. All their blood cells look the same. They all have the A antigen markers. We're going to say these circles here represent the A antigen markers. This person who's heterozygote, you'll notice, has some blood cells that have the A antigen markers, just like before. That's going to correspond to the dominant letter. But the recessive letter is also going to show up, meaning they will have some blood cells that have no markers. The recessive, meaning this lowercase I, actually has no antigenic markers. You can think of it kind of like a naked red blood cell. There's no antigenic markers on it. All right. A person then with, one last thing. Think about it. Would a person who has A antigens on their blood cells have antibodies that flag these red blood cells as bad? So what I'm saying is if a person has A antigens, like we see in the picture up here, these A antigen markers, would their body produce antibodies to tag them as bad? Take a second and think about that. Would someone who has A antigens on their red blood cells, meaning those are their self-antigens, would their body produce antibodies against that blood type? Hopefully you figured this out, but the answer is no. They should not have any antibodies that recognize these blood cells. The reason is those should be marked as self, so the body shouldn't attack those blood cells. They would have, however, antibody B, meaning if a blood cell with B markers comes in, that would be known as foreign. The A antibodies should not be marked as foreign, so they shouldn't have antibodies for them. So let's take a look at a person with type B blood. It's very similar. If they're the homozygous for type B blood, you'll notice all of their red blood cells look the same, corresponding to the two alleles. They all have this diamond shape is what's going to represent our antigenic markers for type B blood. So they all have that same marker. If they're a heterozygote, they have two different types of blood cells. The first one are the ones with the B antigenic markers on it, again. But they also have one for the recessive allele, and if you notice again, it's a naked red blood cell. Just like before, would a person with type B blood want to have antibodies that recognize B antigens? Obviously not, because then they would think their own blood is foreign and start attacking it. So a person with type B blood is going to have antibodies for type A. All right, the last one we have two more. The person with type AB blood is going to have both antigenic markers. If you notice, they have those circle ones that we said represented our A, our type A blood. They also have a second allele indicating the B, which we said is co-dominant, so both are going to show up. So they will have markers marking, they also have antigenic markers that mark their red blood cells as type B. So they have type A and type B. Think about this one. Would this person's body want to have antibodies for type A? Obviously not. They have some A blood, so they wouldn't want antibodies marking their own blood as bad. Would they want to have antibodies for B? No, they would not want to have antibodies for B because also that's going to mark their own blood as bad. So the person with type AB blood actually has no antibodies as far as their blood cells go. All right, then lastly we have our type O blood. This is homozygous recessive, both little i's. All of their blood cells are going to be those naked blood cells like I mentioned. Since they have no antigenic markers, does it matter if they have antibodies for type A blood? That's not going to hurt them. So of course, they will have antibodies for type A blood. What about antibodies for type B blood? That's not going to hurt them either. Since they have no B blood that's their own, sure, they'll have antibodies for that because it's not marking their own as foreign. So type O has both types of antibodies, A and B. turn, based on the definitions we've gone over, try to fill out this chart. Draw in the shapes to fill out each of these things. If you notice, group A wants you to, whoops, sorry. Give me the answers here. Alright, sorry about that. Try this on your own. If you notice group A, what type of blood cells would you find in group A's blood? What type of antibodies would be found in their body based on their blood type? And what type of antigens are visible on their red blood cell? That's going to go right along with this picture here. So try to fill this chart out for people with type A blood, B blood, AB blood, and O. I'll pause, I'll give you some time to fill that out, so go ahead on a piece of paper, draw this chart out, and try to fill out each of these blocks. All right, let's see how you did. Here's how the chart should look. If you notice group A, if it's someone who is heterozygote, meaning they have the I, superscript A, little I, you should have 2 blood cells drawn. If they're homozygous, they're IA, you should have 1 single blood cell drawn. You'll notice I drew the circles for the A antigens. All right, the antibodies they would have in the plasma, they would have anti-B because since they have no A antigens in the body, the anti-B would be there to protect the body from any B cells getting in there since those would be foreign. The antigens on the red blood cell are the A antigens. For group B, it's the exact same thing except with the B antigens. So if they're heterozygote, you should have 2 blood cells, one showing the one with the B antigenic markers, and then the O, which looks like a naked red blood cell. If they're the homozygous dominant, you would have all red blood cells with the antigenic B markers on them. Then the antibodies in the plasma would have to be anti-A, because it's trying to avoid a foreign blood getting into the bloodstream. Foreign blood would be A blood, so it's going to have antibody A. They are marked with B antigens, like we saw. If they're group AB, you should have both antigens drawn on their blood cells. They will have no antibodies, because if they have A antibodies, it would cause a problem to their blood cells. If they had B antibodies, it would cause a problem. So they don't have any antibodies. There's nothing marked as foreign, since they have both types of antigenic markers. We see that they have both A and B antigens on their red blood cells. Lastly, we have group O. This should be a naked red blood cell. They will have anti-A and anti-B, because the body knows that A and B are foreign. It knows the red blood cells are supposed to have nothing on them. If either other type, either the A or the B, gets in the body, it would be considered foreign. So it has antibodies for both A and B. The antigens then are none. Like we said, it's a naked red blood cell. All right. So then the next thing is to talk about transfusions. Your turn. Try to fill in this following chart. Says the people at your table. Of course, you probably don't have people at your table, but try to fill in this chart. If someone has type A blood, meaning they have this type of red blood cell, who can they receive blood from? Meaning, what blood can enter their body and not cause a problem because of the person with type A's antibodies? Looking at the previous chart will be helpful. Also take a look who can type A donate to, meaning what people can accept type A blood. Try that for someone with type A, B, AB, and O. You're probably going to be tempted to fast forward the YouTube video and just get the answers. I'd advise you to please try this on your own because it'll help you out as far as understanding it. Make sure you do understand it. So go ahead and take a few minutes and try to figure this out. All right, let's see how you did. All right, person who has type A blood, they can receive from A, obviously because the blood essentially is the same. They can also receive from O. Remember, type O blood cells don't have any antigenic markers on it. So it's like a naked red blood cell. There's nothing marking it as A or B. You can almost think of it like it's invisible. So they can receive from O. A person who's type B blood can receive from B, obviously for the same reasons. It's essentially the same. They can also receive from O for the same reasons, because O is a naked red blood cell. There's no antigenic markers on it, so there's nothing to tag it as foreign. Someone in group AB can receive from A. Obviously, they don't have antibodies for A, so it wouldn't be harmful to receive straight A type A blood. They can also receive O for the same reasons as before. The O is essentially a naked red blood cell. There's no antigenic markers on it. There's nothing marking it as foreign. They can receive from AB because it's pretty much the same as themselves. And lastly, they can also receive from B. Again, for the same reasons as A, since a person with type AB blood has both A markers, right, A antigenic markers and B antigenic markers, it wouldn't recognize B as foreign because there are no antibodies for group A, AB. Lastly, group O can receive from group O, and that is it. They can't receive from anybody else. Remember, someone with type O blood has antibodies for both A and B, so that eliminates receiving from person A, a person with type A blood, a person with type B blood, or a person with type AB blood. That's why, if you notice, the O, type O can donate to anyone. That's why often the person with type O blood is called the universal donor. Next we have who they can donate to. Someone with group A can donate to someone with type A blood, because again it's the same. Someone with type AB blood, because remember someone with AB blood can have the A antigen, so it's not a problem. Someone with, from type B can donate to someone who has type B, because again it's essentially the same. And also AB for the same reasons as before, because someone with AB blood already has antigenic markers for A and B. So it wouldn't recognize that B blood is foreign. Someone with AB blood can only donate to someone who has AB blood. The reason is because they, if they're donating to someone who has AB blood, that person has, that person can only accept AB because it has antigenic for A and B. Someone in group O can donate to someone with type A blood, because remember, O is like a naked red blood cell. It doesn't have anything marking it as anything, so nobody would recognize it as foreign. They can donate to someone with AB blood, because again, it's naked. Same with B and same with O, because there are no markers on type O. It can basically enter someone's bloodstream without them recognizing it, since it has no antigenic markers. Notice that the AB can donate to anyone, right? Because if someone has AB blood, they don't have any antigenic markers for antibodies for A or antibodies for B, so they can accept anything. That's why a lot of times someone with type AB blood is known as a universal receiver. All right. Quick pedigree review, because in this lab you are going to have to do a pedigree. This should be a really brief review from last year. Remember that the circles are girls, the boys are also represented by squares. The horizontal line means they mated. If there's a line coming down from beneath that, that means they had that person as a child. So this couple, this man and this woman, got together and had this son. This son married this woman, and they had 1, 2, 3, 4, 5 children. That's how pedigree works. You're going to have to fill out a pedigree for some people and identify what blood type they have. Don't forget, we have 6 genotypes for 4 blood types. There's the big I, a little I, the heterozygous for type A blood, or the homozygous for type A blood. There's the same 2 for type B blood. Then there's type AB blood. There's only 1 genotype for that, and there's also 1 genotype for type O. So remember, divide those up, we have A, B, AB, and O. You're going to have to identify, based on your results of this blood typing lab, if a person's AB, or O, and you'll have to figure out their pedigree based on that. One other thing I want to briefly cover that I don't believe I have in this PowerPoint. So let's pull up a blank page on a flip chart. The last thing is, you've probably heard of people being AB positive or AB negative. So let's quickly talk about what that positive and negative means. It's the exact same thing as the other markers, right? So here's a red blood cell. Here's a red blood cell. There are those markers. The plus and minus stands for an RH marker. So we'll just make it really obvious and make the shape of my RH an RH. If someone has RH, that marker, that antigenic marker, would consider them RH positive. If their red blood cell does not have that RH marker, they're RH negative. Don't be confused, this is the same thing as the A and B, right? Just like if someone has an A marker, where we have circles before, someone has an A marker, we'd say they are type A. If they have the A marker and an RH, we would consider them A positive. If they're type A and don't have the RH, they're A negative. Going right along with that, if they are, what else do we have? The diamond. That's type B blood. They can have that with the RH marker, and they would be B positive. Or they can have their B marker and not have the RH, and that would be B negative. So it works the same way. The positive and negative is just one particular antigenic marker. That should cover everything you need to know for this lab. The lab is going to ask you to blood type different people. It's pretty well explained how to do the lab in the lab. And if you're doing the lab without me, so for period 2 and 3, I'll have a worksheet, an additional little supplement to help you figure out this lab on your own. If you're at all confused, you will be with me again on Thursday, or you can swing by any time you want. Swing by the classroom later on in the day after the Keystones are over, and I can try to help you out. Or, like I said, you'll have me on Thursday. You can also attend the supplemental or the replacement lab, whatever you want to call it, for the PM activity period on Thursday. That should cover everything. If you have any questions at all, please let me know.